Alright, so this is part 2 of, I forgot what this was, of section 10.3. We were just talking about translation, and we're moving on to, what is this? Transfer RNA. tRNA and anticodons. Amino acids floating freely in the cytosol are transported to the ribosomes by tRNA molecules. Alright, so I had trouble understanding what tRNA molecules were, and now we're seeing that they transport the amino acids around the cytosol. In figure 10.8, a tRNA molecule has a region that bonds to a specific amino acid. Alright, so, oh, there it is. I'm gonna put this down for a second. I have to do a little bit of reading before I kind of understand what that figure is. Anyway, notice also that the loop opposite of the side of the amino acid attachment bears a sequence of 3 nucleotides called an anticodon. So, okay, I'm gonna hold this up and try to explain it to you. It's a little difficult to have all this. Okay, so as you can see, this is the... Alright, hopefully you can see it there. But, this is not working. Okay, I'm gonna put it underneath right here. Alright, so right here, these are the anticodon. These right here are the hydrogen bonds, and this is the site of attachment to the amino acid. Now, I'm not exactly sure how all this works yet, so please bear with me. The caption here says that each transfer RNA transports a specific amino acid to the ribosome during translation. So this happens while the messenger RNA is taking the information to the ribosomes. So this is getting a little bit complex, if it hasn't already. The transfer RNA anticodon is complementary to and pairs with its corresponding mRNA codon. So we know that codons are made up of nucleotides, and nucleotides all have base pairs. Now it's saying that these groups of 3 nucleotides also have base pairs. So a codon would pair with an anticodon. Notice that in figure 10.9, the same base pairing rules followed during transcription. Now, transcription again is the reading of like adenine, uracil, guanine, adenine, and all of that in the transcription of it from DNA to RNA. Where was I? For example, a transfer RNA with an anticodon sequence of AAA, adenine, would bind to the messenger RNA codon sequence of UUU. So I'm seeing from this that the anticodon would kind of be like the DNA in a sense that it has the adenine part of it, and the codon would kind of be like the RNA because it has uracil. The sequence would be specific for the amino acid. Oh, I'm gonna mess this word up. Phenylalanine. Thus, the pairing of an anticodon with the with a codon ensures that the amino acids are added to the growing polypeptide in the order prescribed by the messenger RNA transcript. Now, a polypeptide is just the product. Don't be confused. A polypeptide could be a protein. The polypeptide could be something else. Ribosomes. Ribosomes are composed of rRNA, which is, if you will, hold on, ribosomal RNA. Ribosomes are composed of ribosomal RNA and proteins that are usually both free in the cytosol and attached to the endoplasmic reticulum. I'm going to call that the ER because it's a bit of a mouthful. Ribosomes that are free in the cytosol, as shown in figure 10.9, produce proteins that will be used within the cell. So these freelance ribosomes are making these proteins that are going to function in the cell. Membrane proteins and proteins that will be exported for the use outside of the cell are produced by ribosomes attached to the ER. So the freelance ribosomes produce proteins that are used in the cytosol. So the ribosomes are in the cytosol, so the proteins are going to stay in the cytosol. And if you guys know, the ER kind of like ships everything outside, so it makes sense that ribosomes attached to the ER are going to get shipped outside of the cell. Ribosomes have 3 binding sites that are key to translation. Again, translation occurs while messenger RNA is taking information from the nucleus and putting it into the cytosol. One binding site holds a messenger RNA transcript so that its codons are accessible to the rRNA molecules. The other 2 binding sites hold transfer RNAs whose anticodons paired with messenger RNA codons. Okay, that seems pretty complex, but as we already went over this, the transfer RNA has anticodons, and the anticodons get paired up with certain messenger RNA codons. So the anticodons would be the nucleotides would have to match up with the codons. So if it's adenine, 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 it would be uracil, uracil, uracil. If it's guanine, 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 it would be cytosine, 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 cytosine. Okay, this is the last part of it. Protein assembly. The assembly of a polypeptide begins when a ribosome attaches to the start codon on a messenger RNA transcript. Find the start codon on the messenger RNA transcript shown on figure 10.9. Okay, well, it's a long strand of signals, and I'm gonna have to find a certain combination, so it's nothing that you're gonna miss. The start codon pairs with the anticodon UAC. So the start codon is AUG, and it would pair with, oh, my cat just walked in, sorry, with UAC on a transfer RNA, because the transfer RNA that bears the UAC anticodon, so the transfer RNA has the anticodon and the messenger RNA has the codon. The UAC anticodon also carries the amino acid methionine. The first amino acid in every polypeptide is initially a methionine. So I believe the methionine is the beginning of the sequence. It's the very first codon to go on to the other codon. So the start codon would be right here, and then all these other codons with all the other information be in between. And then at the end would be the stop codon. So that is pretty basic. You start, and then you have all that, and then you stop. I'm almost out of time. I just want to finish this off. As a ribosome moves around the messenger RNA transcript, each messenger RNA codon is sequentially paired with its transfer RNA anticodon. As figure 10.9 shows, the pairing of an anticodon with a codon causes the specified amino acids to attach to the previously translated amino acid with a covalent bond called a peptide bond. So the product is a polypeptide, and this covalent bond would be a peptide bond. In this way, amino acids are joined to a growing polypeptide chain in the order specified by the messenger RNA transcript. So this is all happening under the messenger RNA. I need to finish this last part in the next movie because I am out of time. So thank you for listening.